So, good evening to all. Welcome to week 9 tutorial session of experimental biotechnology. I'm Ananya Chatterjee, a PML scholar and PhD student in IISc Bangalore. I discuss weekly course on experimental biotechnology, and course instructor for this course is Professor Vishal Trivedi from IIT Guwahati. So, so far we have discussed till week 8 tutorial sessions, and today we'll be starting from week 9, which is molecular biology techniques. And so far we have discussed assignments 1 to 7 from current assignment as well as from previous year assignments. And every tutorial sessions class I upload in this YouTube link. You can access it through NPTEL website, and every lecture slides I upload in this folder. So today's class I'll be starting with from week 8 assignment solve, and then I will proceed to week 9 content. So if I'll be back in 5 to 6 minutes, please be ready with, if you have any doubt, put down in the comment section and I'll be checking, and after 5 minutes I'll be starting with discussing week 8 assignment. Hello all. Hi. Sorry for the late. We'll be starting today's class of week 9 experimental biotechnology. So far we have discussed till week 8. Today we'll be discussing week 8 assignments which you were given. So the first question is, what is the major structural difference between prokaryotic and eukaryotic cells in terms of their DNA? Option 1, prokaryotic cells have linear DNA while eukaryotic cells have circular DNA. Prokaryotic cells have multiple chromosomes while eukaryotic cells have a single chromosome. Prokaryotic cells lack DNA, eukaryotic cells have DNA in the form of plasmids. Prokaryotic cells have a single circular DNA molecule while eukaryotic cells have multiple linear chromosomes. So the correct answer should be answer D. Okay. So far till week 8, if you have any doubt, you can comment below in comment section. So now I think I am audible and visible to you. So second question is, carbohydrates in growth medium is used for what in the cells? So in the growth medium, why do you put carbohydrates? Options: energy generation, nucleotide synthesis, amino acid synthesis, and prokaryotic synthesis. We know that carbohydrates are required for glycolysis, right? So for that we need for it, the reason is for energy production, or energy generation. Question number 3, which of the following is an example of a defined or synthetic media? So previous day's class, we have discussed those. There was one chart where we listed down some synthetic or defined media. So if you follow that chart, you will find that defined or synthetic medium is minimal salts media. Okay. What is added to DMEM mammalian cell culture as a buffering agent? Sodium bicarbonate, ammonium sulfate, sodium phosphate, potassium phosphate. So what would be sodium bicarbonate? Okay, for pH balance or buffering system. What is added to DMEM mammalian cell culture media as a growth factors? For growth factors, we know serum is added. So for serum, what is the source? Fetal bovine serum, FBS, also called FBS. FBS. So mammalian cell culture medium is filtered and not autoclaved because the glucose present in the media might get charred. The growth factors are heat sensitive and might get degraded, which is true. Filtration is cheaper, no. Media components react with each other inside the media, no. So the answer will be the growth factors which you use, like FBS, it has proteins, right? So proteins are heat sensitive, so might get degraded. That's why autoclave means what? 120 degrees Celsius we have to heat, and we can't afford heat FBS at 120 degrees Celsius. That's why we do filtration. Why is pH meter not used for checking the pH of DMA medium? So what would be the reason? Media components might get blocked the pH probe bulb. Media might get contaminated. pH meter doesn't work inside DMA media, which is also not true. pH drives are more precise than pH meter, which is not true. Okay, this is not true. This is not true. Contamination, it might get true, but but this is not the reason, because you can be sterile while making. You can remove the contamination issue, but the main reason is that whatever media components are there, that might block the pH probe bulb. So the answer is this. Why is serum heat inactivated before putting in cell culture media? So there are some complements are there in the FBS. So to inactivate those complements, we do heat inactivate before putting. So heat inactivation temperature is around 50 to 60 degrees Celsius. What is a media is used for production of baculovirus in Lepidopteran cell line? So you have to remember this, it was TC100 media. So there was one chart in last year's last classes session, so there it was written. Which of the following is a characteristic feature of eukaryotic cells but not prokaryotic cells? Okay, presence of ribosomes, nucleus, cell wall, circular DNA. So answer will be definitely nucleus. So prokaryotic cells have nucleoid, but eukaryotic cells has nucleus. What allows for mobility of prokaryotic cells? Flagella. The region between inner layer and outer layer of gram-negative prokaryotic cell membrane is called periplasmic space. What is the role of phenol rate present in mammalian cell culture media? To monitor pH of the media. Okay. In gram-negative bacteria, purines are present in where? Outer membrane. Bacterial cell wall is composed of peptidoglycan. We all know these answers. Now today I will be starting with molecular biology, week 9 content. So these slides are from Professor Vishal Trivedi, which you also see in NPTEL. So we will be discussing these slides. If you have any doubt while you went through, you can let us know. So we will be starting with today's discussion on polymerase chain reaction or PCR. So some of you might have done PCR in your lab. Some of you might have just went through the theory portion. So we'll be discussing today what is PCR. So PCR or polymerase chain reaction. So what do you suggest, like what this name suggests? Polymerase means polymerization of something. Okay. Chain reaction. Reaction means something reaction is happening. Okay. So this is something which deals with replication, like something you are replicating or polymerizing. Okay. So what happens in PCR, you amplify a lot of double-stranded DNA molecules or fragments with same size and sequence by enzymatic method and cycling conditions. Okay. So in polymerase chain reaction, you have DNA segment, okay, and you will have some primers, so that from those primers, those replication will initiate. So you need DNA. DNA is a nucleic acid that is composed of two complementary nucleotide building block chains. So it has, we know that's helical, two helix, those are complementary. The nucleotides are made up of phosphate group, a 5-carbon sugar and a nitrogen base. DNA has 4 nitrogen bases, 2 are purines, 2 ring base adenine and guanine, and 2 are pyrimidids, cytosine and thymine. These 4 bases are linked in a repeated pattern by hydrogen bonding between the nitrogen bases. The linking of the two complementary strands is called hybridization. DNA is complementary in nature. So, this is a primary strand, 5' to 3', and against that you have another strand of 3' to 5'. Okay, so this is complementary to this 5' to 3' strand. So DNA synthesis is linked to cell division and growth. So here in your cell you have plasma membrane, cell wall, and duplication of chromosome happens. This is continuation of growth of cells, and after cell division, cell divides into 2 cells. So cell cycle are composed of G1, S, G2 and M phases. So DNA duplication, process of duplication of the entire genome prior to cell division. Biological significance, extreme accuracy of DNA replication is necessary in order to preserve the integrity of the genome in successive generations. In eukaryotes, replication only occurs during the S phase. Replication rate in eukaryotes is slower, resulting in a higher fidelity or accuracy of replication in eukaryotes. DNA replication is semi-conservative, starts at the origin of replication. Synthesis by DNA polymerase. So DNA polymerase is the enzyme which helps in DNA replication, and synthesis happens in the 5' to 3' direction. It can be unidirectional, it can be bidirectional, semi-discontinuous because of production of lagging and leading strand, and RNA primers are required. RNA primers means single stranded primers are required. DNA replications are 3 steps event. So initiation, that is prepare of DNA for synthesis. Proteins bind to DNA, opening of double helix DNA, elongation, synthesis phase. Proteins connect the correct sequences of nucleotides into a continuous new strand of DNA. So elongation means synthesis phase happens. So and termination, stopping of DNA synthesis. Protein release the replication complex. So if enzymes or proteins are getting released, then the reaction will also get stopped. DNA replication, helicases. So what are the enzymes required for DNA replication? Helicases, primase, SSB protein, DNA polymerase, tethering protein. Helicase means separates, so helicase basically separates two strands. Primase, RNA primer synthesis. SSB protein prevent reannealing of single strands. DNA polymerase synthesis of new strand, and tethering protein stabilizes polymerases. So what is the function of helicase? Helicase basically separates two strands of DNA. Primase RNA primer synthesizes RNA primer, which is required for priming and then starting the replication. SSB protein, this is single stranded binding protein, so it prevents reannealing of single strands. DNA polymerase helps in synthesis of new strand, and tethering protein stabilizes polymerase. So in the case of DNA synthesis, initiation, that means preparation for DNA to be synthesized. Elongation, that is synthesis phase. Termination, stopping of DNA synthesis. Development of polymerase chain reaction. So different events in development of PCR. The gradual breakthrough from the discovery of DNA structure to the invention of modern PCR. Discovery of mechanism of DNA replication by Arthur Kornberg. He discovered the first DNA polymerase and other factors like helicase and primase. In 1976, so what happened? Isolation of thermostable DNA polymerase from T. aquaticus, Thermophilus aquaticus. In 1983, Mullis synthesized DNA oligoprobes from sickle cell anemia mutation. 1983 again, repeated thermal cycling was first used for small segment clone gene. 1984, Mullis and Tom White designed experiments to test PCR on genomic DNA, but the amplified product was not visible in agar cell. 1985, patent was filed for PCR and its applications focusing on sickle cell anemia mutation. 1985, the use of thermostable DNA polymerase in PCR was started. Out of only two enzymes, TAC and BST, known at that time, so TAC was found more suitable for PCR. 1985, first announcement of PCR techniques in Salt Lake City, and 1985 and 1987, development of instruments for PCR and its reagent. Now we'll discuss PCR. So PCR is a repetitive cycle reaction that involves mechanism of DNA replication. It results in production of multiple copies of DNA from a single one. The whole process involves three main events: denaturation, annealing, and elongation. A DNA fragment of interest is used as a template from which a pair of primers or short oligonucleotides complementary to the both the double strands of DNA are made to prime the DNA synthesis. So for that, what we need some primers. We need complementary primers so that those come and bind to one of your strand and then start the replication. So at first, what we have to do, we have double stranded DNA, but we need to denature those. So we'll denature those, so it will be in two single stranded. Now we'll add primer. So we have to induce primer binding. So whenever the primer will get bind, it will start elongation and DNA will, like the whole double stranded DNA will be produced. So we'll do it in 1 cycle, 2 cycle, and 3 cycle, and so on. Generally around 25 to 30 cycle people give in PCR reaction. So DNA synthesis, where the direction of synthesis or extension is from 5' to 3'. So always 5' to 3' the direction will be. The number of amplified DNA or the amplicons increases exponentially per cycle. Thus, one molecule of DNA give rise to 2, 4, 8, 16 like that. So what is the formula? Amount of amplified DNA C equals to C naught. C naught is the initial amount of DNA. C naught into or multiplication 1 plus E. E is efficiency to the power n. Okay, N means number of cycle. Polymerase chain reaction. So at first, so there is some stages: stage 1, stage 2, stage 3. So at first, initial denaturation, you have to heat the PCR mixture around 94 to 96 or 98 degree. So this is denaturation step or initiation, like initiation step. You have to now heat it. Then stage 2, you start the cycle from denaturation itself. So this is the first step in which double stranded DNA template is denatured to form single stranded DNA by heating 95 degrees Celsius for 15 to 30 seconds. This varies, 95 or 98 also you can use. So you have to give it for like 15 to 30 seconds. Then annealing. Annealing means this is a temperature, this is a step where at low temperature, usually 50 to 65 degrees Celsius, primers are allowed to bind to template DNA. So at this annealing temperature, primer will attach to the complementary strand. After cycles, now you will have elongation step. So elongation step means this is a synthesis step where the polymerase performs synthesis of new strand in the 5' to 3' direction using primer and DNTPs. An average DNA polymerase adds about 1000 base pair per minute. Step 1 to 3 makes one cycle, and in general 30 to 40 such cycles are performed in a typical PCR application. So after the cycles are completed, the reaction is held at 72 degrees Celsius for several minutes to allow the final extension of the remaining DNA to be fully extended. So whatever, like while replication, whatever is it started replication, but some of the amount is still need to be replicated, so it gives time to extension. And then final hold at 4 degree means your PCR reaction is done, and you have to just take out your PCR reaction. You have to keep for short term storage, you have to keep at 4 degree, and for long term storage you can keep at -20. So PCR. So the steps again: initial denaturation step around 95 to 90 degree for 5 minutes. Thermocycle file which is around 30 cycle. Denaturation 95 degrees Celsius for 30 seconds. Annealing 55 degrees Celsius for 30 seconds. Extension 72 for 45. Final extension 72 for 5 minutes. Holding for usually at 4 degree. Requirements for PCR: DNA sequence of target region must be known. Primers you need to have typically 20 to 30 bases in size. This can be readily produced by commercial companies. Can also be prepared using DNA synthesizer. Thermostable DNA polymerase, example Taq polymerase, which is not inactivated by heating to 95 degrees Celsius. DNA thermal cycler, machine which can be programmed to carry out heating and cooling of samples over a number of cycles. So you need a PCR machine, so which is also called thermal cycler, because it's programmed to carry out heating and cooling like in a cyclic manner. So this is basically an old PCR instrumentation system. So nowadays also you will be able to see like this, only same thing. This is the heating block. So here your temperature will change, so you need to put your sample here, and you need to close the door, and here your parameters will come up, where you will select those like you need to select your protocol, how much temperature and for how much time you want to set, and you will start the run. So this is a thermal cycler. So it's the instrument that carries out the amplification via polymerase chain reaction. This device has a thermal block with holes where tubes holding the reaction mixture can be inserted. The cycler then raises and lowers the temperature of the block in discrete pre-programmed steps. Instrumentation: most conventional thermocycles rely upon Peltier effect, thermoelectric heating and cooling. The thermoelectric module is composed of micro modules connected in series. Each micro module is composed of N and P type T semiconductor material. The Peltier phenomenon occurs at the junction of these two dissimilar conductors. When current is passed through the junction, a transfer of heat occurs from one side of the junction to other. QL is the absorbed heat on the cold side, and QA is the released heat on the heater side. So it based on Peltier effect, thermoelectric heating and cooling. So this is a heating plot. Here this is sample block, and you have vessels. So here you will keep your sample and DNA will be there. So this is hot side, this is T module. So T semiconductor material, basically this is cold side, and here heat exchange happens. So template DNA around 1 picogram to 1 nanogram for viral or short templates, 1 nanogram to 1 microgram for genomic DNA. Primers around 0.5 micromolar for each primer, forward and reverse primer. Magnesium chloride you need to give around 1.5 to 2 millimolar. DNTP's typical concentration is 200 micromolar of each DNTP. Tag polymer is 0.5 to 2 unit per 50 microliter reaction. Template DNA, genomic DNA, DNA fragment, plasmid, viral and tissue sample. Primers. A primer is a short DNA trace that serves as a starting point of DNA synthesis. In PCR, 2 primers are required to bind to each of the single stranded DNA obtained after denaturation, flanking the target sequence. So you need 2 primers, basically forward and reverse. So your DNA is like that. So you need 1 primer to synthesize this strand forward and 1 primer to synthesize this strand reverse. Okay, the primers have a sequence complementary to the sequence. So this has complementary to this, and this primer has complementary to this sequence in the template DNA where they are supposed to start synthesis. So TAG stands for Thermus aquaticus, which is a microbe found in 170 degree Fahrenheit hot springs in Yellowstone National Forest. Okay, this is pretty cool to understand. So TAG DNA polymerase is stable in high temperatures and acts in the presence of magnesium. The optimum temperature for TAG polymerase is 72 degree. TAG polymerase lacks 3' to 5' exonuclease proofreading activity commonly present in other polymerase. TAG misincorporates one base in 10 to the 4, so there is some error rate. A 400 base pair target will contain an error in 33% of the molecules after 20 cycles. Error distribution will be random. So alternatives, you can use PFU DNA polymerase from Pyrococcus furiosus, possesses 3' to 5' exonuclease proofreading activity. The error rate is only 3.5% after 20 cycles. More amount of primer is added to avoid primer dimering. So for unexplored genes, primers used in closely related spaces are used. Okay. So this is PCR reaction setup. You have to be ready with template DNA, primers, magnesium chloride, DNTPs, tag polymerase, buffer, water, and total volume can be 20 microliter, 25 or 50 microliter. Okay. So and then in that display mode, you have to set up your protocol. So you have reaction cycles, you have primers, enzymes, additives. So after your experiment, you need to do some optimization if it doesn't come well in one go. So if no product or carrier size product produced, so what you do after PCR amplification, you will take out those samples from those thermal cyclone and you will run the gel. In the gel, you need to observe your desired band. So if you don't see any band, or if you see a different size, then you have to check at first DNA quality. You have to reduce annealing temperature, have to increase magnesium concentration, add DMSO around 10%, use different thermostable enzyme throughout primers, make new stock. So you have to again design the primer and do the experiment. If extra spurious product bands are there, increase annealing temperature, reduce magnesium concentration, reduce number of cycles, try different enzymes. So these are the different troubleshooting which will help you during PCR. Now analysis of PCR reaction. So once PCR cycle is completed, the amplified product is loaded into the agarose gel and observed after yttrium bromide staining under UV light. A water blank reaction is included to monitor the cross-contamination DNA source as template. The percentage of agarose gel depends on the size of DNA to be visualized. Generally 0.8 to 1% agarose gel is used for 0.5 to 5 kV amplified DNA, while a DNA of larger size or genomic DNA is visualized in a gel as low as 0.5%. So these are the markers this is negative amplification, where you can see template DNA, and this is positive amplification. So you have seen amplified DNA of interest, and in the negative amplification you have seen primer dimer at very lower end of agarose. Advantages of PCR. So it speeds up your reaction, ease of use, sensitivity is higher, and robustness. Limitation of PCR: need for target DNA sequence information, primer designing for unexplored one, boundary regions of DNA to be amplified must be known. Infidelity of DNA replication: tag polymerase, no proofreading is there, so around 40% after 20 cycles. Short size and limiting amount of PCR product. Up to 5 kb can be easily amplified, up to 40 kb can be amplified with some modification, cannot be used in genome sequencing project. Now the instrumentation: thermal cycler is the instrument that carries out the amplification via polymerase chain reaction. The device has a thermal block with holes where tubes holding the reaction mixture can be inserted. The cycler then resists and lowers the temperature of the block in discrete pre-programmed steps. So far we have discussed PCR. Now we'll be starting discussing application of PCR. So we'll be starting with application. Analysis of PCR reaction we have covered. We'll starting with application of PCR. So there are few applications of PCR. Molecular identification, in the case of molecular archaeology, molecular epidemiology, molecular ecology, DNA fingerprinting, classification of organisms, genotyping, prenatal diagnosis, mutation screening, drug discovery, genetic matching, detection of pathogens. In the case of sequencing, bioinformatics, genomic cloning, human genome project, genetic engineering to do site-directed mutagenation, gene expression. So there are very varieties of utilities of PCR. PCR in food science. PCR is a rapid and sensitive method that enables detection of subdominant populations in food without the need of enrichment of media. It allows detection of dead cells and non-cultivable cells. So applications of PCR in food microbiology. There are several microorganisms: Salmonella, different species of Salmonella, Listeria, monocytogenes, Staphylococcus, Enterobacteriaceae, Escherichia coli, Bacillus. And you have some target genes, right? So those you can target those target genes, and applications will be detection, quantification. Okay. So test characteristics: enrichment plus qPCR, TACMAN, enrichment per multiplex qPCR. So by this qPCRs and RT-PCR you can be able to detect those targets. So inference: artificially contaminated chicken meat, minced meat, salmon, raw milk, artificially contaminated chicken, naturally contaminated chicken, artificially contaminated beef. So this test samples you can be able to do, and you can be able to detect those target genes. PCR in medical science. The use of PCR in medical science already began in 1985, and a viable test of measuring amount of HIV in the blood was published in May 1987 by John Sinsky at CITES. In 1989, Norm Arman and team developed multiplex PCR. An application for single cell was performed for the first time on single sperm cells to directly analyze the products of meiotic recombination, and was also applied on the other targets like human leukocytes antigens. PCR technology has become an essential research and diagnostic tool for improving human health and quality of life. It allows the detection of infectious organisms just from one cell by amplifying specific regions of the genetic material. So PCR in infectious disease. So PCR technology has become the basis for a broad spectrum of clinical diagnostic tests for various infectious diseases including viruses and bacteria. Unlike the traditional antibody-based diagnostic tool that depends on the patient to develop antibodies over time, PCR allows faster diagnosis which hasten the treatment and recovery. Besides detecting the presence of pathogens, PCR allows us to quantify the amount of pathogens present in patient's blood. What does it mean? That if you are infected with some virus, okay, so previously what is the way that you need to wait for some time and your blood will be collected and yours from serum you will check that whether antibody against those pathogens are there. But if you want to check it early, what you will do, you will collect your sample, blood or saliva or whatever, and there you will do PCR or RT-PCR. So what will happen if you have your specific pathogen is there, that will be amplified. So from there you will be able to detect like HIV-1 which causes AIDS, hepatitis B and C, human papillomavirus, chlamydia, Neisseria, cytomegalovirus. This all pathogens you will be able to detect. PCR in infectious disease. So you will collect your blood, then you will do PCR and analyze in gel. PCR in blood screening. PCR technology helps in detecting the presence of infectious disease in the donated blood samples. Benefits of using PCR technology to monitor the world's blood supply include a decrease in the waiting period during which the infectious agent is undetectable by serological screening technologies that rely on the formation of antibodies. So you don't have to rely on and wait for generating the antibody. So you can directly amplify the pathogen present on your body and you can do the PCR and detect those. So the ability to perform combined blood screening for common life-threatening pathogens like hepatitis B, hepatitis C and HIV-1. Combination of PCR-based tests incorporated into blood donor screening programs could increase awareness and reduce the risk of virus transmission. PCR in plant science. So there are various fields in plant science which requires the use of PCR technology for its accomplishment. Plant species identification. PCR technique has also been employed in identification of plant species using species and groups of specific primers targeting chloroplast DNA. This assays always identification of plants based on size specific amplicons. For example, plants belonging to the same family has close primer binding sites and hence some amplicon size, while plants belonging to the different species and groups have different primer binding sites, hence will result in different amplicon size. Plant microbe interaction. So there are several pathogens: Clavibacter, Ralstonia, Acidophorus, Argobacterium, and so on. So and they happen to be infecting different plants. So RT-PCR techniques, due to its rapidity and sensitivity, has also seen its uses in detection of microbes investing a crop plant. Early diagnosis of the pathogens is essential to provide rapid and suitable measurements for limiting the epidemics and selection of appropriate control measures. PCR in tissue culture. So the use of PCR in tissue culture was already reported in 1992. It was used in analysis of DNA and specific genes in plant cells at different stages of regeneration during in vitro culture, along with random amplification of polymeric DNA. The level of polymerization is regenerated plants could be revealed by these dual techniques. PCR could flawlessly amplify neomycin phosphatase transferase gene, an antibiotic which is used as selective marker in transgenic plants. PCR in veterinary parasitology. So there is some disease, pseudorabies virus of pigs. The virus causes abortion and mortality in piglets. So this disease has a latent period where there is no symptom of infection, making it difficult to eradicate the disease completely. For this reason, PCR is considered to be appropriate tool for detecting latent cases. Bovine leukemia virus. The virus causes engenetic bovine leukosis. PCR assay of detection of VLV was developed. Bovine viral diarrhea. The virus is not only fatal to cattle but also causes contamination in calf serum. used in cell culture. Besides the above example, PCR has been used in routine diagnosis of veterinary viruses such as porcine parvo, bovine papilloma type 1, avian papilloma, and so on. Also, you know that PCR is very much important in forensic science, in criminology, in detecting parentology and all. So individuals has different DNA profiles known as DNA fingerprinting. Okay. A DNA fingerprinting uses variable number of tandem repeats loci, as this loci is so variable that unrelated individuals are unlikely to have the same VNTR. So in crime scenes DNA, and if you are detecting with suspect 1, 2, 3, and 4, one you is getting matched, so this is the suspect, 3 is the convict. So you can do PCR and you can check that those are matching exactly. So parental testing also you can do. So PCR technology is also used in finding the biological parents of adopted or kidnapped child, where the DNA of a child is matched with close relatives. So the actual biological father of a newborn can also be ruled out. In parental testing, short tandem repeats are used as marker, where each person's DNA copies contain 2 copies of these markers, one each from father and mother. These markers differ in length and sometimes sequence. Another sensitive technique that can be used to establish maternal relationship between people is called mitochondrial DNA analysis, which relies on PCR. This analysis is better than fingerprinting for samples which become too old that the nucleus of the cells gets degraded. Okay. So in DNA markers A, B, C, D, E, mother 2631, child has 26 and 30, father has 29 and 30. Okay. So like that, one is coming from father and one is coming from mother. PCR in research application. DNA cloning, if you want to clone a gene and then further you want to do transfect and then protein expression, you need to do DNA cloning, right? So PCR helps to amplify specific DNA from a genome, and the amplified DNA can be inserted into a vector for transformation and expression. These inserts can further be confirmed by PCR method. DNA sequencing you can do. Sequence tag size. So there is process where PCR is used as an indicator if a particular segment of a gene or genome is present in a particular clone. This application is vital in mapping the cosmic clones. Phylogeny analysis. The phylogeny of organisms like plants, animals, and other local organisms can be traced out by DNA analysis. The DNA of different organisms can also be distinguished. For example, in mammals, the conserved regions such as 3' UTR of the SON gene can be used to identify the mammalian origin of a sample. Using this test, the DNA of a human, monkey, cat, dog, mouse was distinguished. Gene expression studies. Tissues can be analyzed at different stages to see which genes have become active or switched off. The level of protein expression can be studied by quantifying the gene using qPCR. So polymerase chain reaction, you don't have the sequence, you have genomic sequence, you will do PCR, you will do fragment and then cloning. So in polymerase chain reaction, if you have the gene sequence known, so for that you need to, so gene sequence need to be known because you need to generate primer. So from that you will do PCR and then you will proceed for cloning. Okay. So there are various uses you can see. So uses of PCR in forensic science, in research applications, in veterinary parasitology, in tissue culture, in plant microbe interaction, in plant science, in blood screening, in infectious disease screening, in medical science, in food microbiology, in food science. So these are various important uses of PCR. If you have any specific doubt, we'll discuss. So we have discussed week 8 assignments. If you have any doubt in week 8's assignments or any assignments, you can let me know. I will discuss in today's class or in next class. So Any doubts so far? So I think we have three classes left, tutorial session 10, 11 and 12. So now molecular biology techniques are going on. So next class I will meet on Tuesday again from 6 to 8 p.m. And your assignments are also live for week 9 and week 10. So please go through those assignments. If you face any problem, just let us know in the today also. You can let me know in the comment section, or you can mail us in those discussion forum, so that we'll be getting back to you and then discuss. So far we have discussed till week 8, and today we have completed week 9 session, and we have discussed till assignment 8. Today we have discussed assignment 8, and tomorrow, like next class, I'll be discussing on assignment 9. So if you don't have any questions so far, we'll stop today's class. Next, again, we'll meet on coming Tuesday from 6 to 8 p.m. We'll be discussing if you have any doubts from your assignments. So far, we have discussed till assignment 8. Next class, we'll be discussing assignment 9 and 10. And we'll be also going through those lecture slides by Professor Vishal Trivedi. If you have any doubt, we'll be going through. So, thanks to all. We'll be stopping today's class and bye.